Hi everyone, thanks for joining us. Today I'll be talking about the nature, ecology, and evolution of egg color. Birds are the only living animals that lay colored eggs, and the diversity of color and pattern in their eggshells is quite remarkable, as you can see here from my background photograph. Colors range from bright blue to brown, and we have all kinds of spots and speckles present. All this diversity in color and pattern traces back to only two pigments, called protoporphyrin and biliverdin. Protoporphyrin is a red pigment and biliverdin is a blue pigment. And the overall color impression in eggshells results from the way they are mixed within the eggshell. Protoporphyrin and biliverdin belong to a class of compounds that is called tetrapyral pigments, based on the four pyrrole units that make up their molecular skeleton. So you can see here, 1, 2, 3, 4. Protoporphyrin is closely related to heme, which is the compound that gives the red color to our blood. And even though protoporphyrin and biliverdin look very similar in their molecular structure, there's a small difference. This methane bridge here in protoporphyrin is cleaved in biliverdin and replaced by these two carbonyl functions. That determines not only the complementary colors that they produce as pigments, but it has also a huge influence on their solubility properties. Protoporphyrin is much more fat soluble than biliverdin, which is rather water soluble. These differences in their solubility are thought to explain why biliverdin and protoporphyrin are incorporated in so fundamentally different ways in the avian eggshell. Here we are looking at a schematic drawing of a cross-section of bird eggshells. Here on the left side, we're looking at a blue eggshell that contains biliverdin. And here on the right side, we're looking at a brown eggshell that contains protoporphyrin. As you can see here, biliverdin occurs in highest concentrations in the organic waxy cuticle that sits on top of the mineralized part of the eggshell. Biliverdin occurs also in the mineralized part of the eggshell and extends here into the deeper parts of the prismatic zone. Protoporphyrin is restricted to the waxy cuticle on the top, where it is dissolved in the fatty waxy phase. However, protoporphyrin extends minimally into the mineralized part of the eggshell. Contrary to biliverdin, protoporphyrin can produce spots and speckles. If we have different concentrations of protoporphyrin here in the waxy cuticle, we end up with some sort of spotty pattern. Biliverdin can only generate some sort of background coloration if it occurs together with protoporphyrin. Our latest research suggests that the differences in the distribution of biliverdin and protoporphyrin are not only based on the differences in solubility, but also on the way the pigments interact with the eggshell proteins that sort of form the organic skeleton of any kind of eggshell. And you can see this basically here in this figure, where we're looking at real eggshell cross-sections of different birds and their demineralized residues. Demineralized means basically that we've removed all of the eggshell mineral with acid treatment, and we're looking at what is left over. As you can see here in this emu eggshell, biliverdin extends into the deeper parts of the eggshell and is bound to the protein phase of the prismatic zone and also the cuticle. Protoporphyrin, on the contrary, is restricted to the uppermost part here of this chicken eggshell and is primarily stored in the waxy cuticle, the orange one that you can see here. In a spotted eggshell, like for example this tern eggshell, we have different concentrations of protoporphyrin in the waxy cuticle, and these local accumulations eventually generate spots and speckles. It has long been thought that egg color is some sort of evolutionary invention of modern birds, and that birds potentially evolved egg color multiple times independently. But as paleontologists, we know that many traits that we thought that are unique to modern birds actually evolved in their dinosaur ancestors. All birds are dinosaurs. Here we're looking at an archosauromorph tree of life. And so when we think about egg color as a potentially inherited trait, then we have to find a starting point here in our archosaur tree of life to figure out where we have to start analyzing eggshells for potentially preserved color pigments. And to do so, we came up with a really simple hypothesis, saying that once we have an avian-style open nest in which eggs are exposed to the open, exposed to parents, exposed to predators, there should be some sort of evolutionary, like selective force to evolve egg color. And with this idea, we sampled eggshells across our Archosaur tree of life. Figuring out the nesting mode in fossils is quite easy for one simple reason. All eggshells have pores. The pores sustain the respiration of the developing embryo inside. Eggshells that are buried underground have more pores than eggshells that are stored in the open, simply because the soil is sort of preventing diffusion of oxygen. So the embryo needs more pores to get enough oxygen to develop properly. Based on these eggshell porosity measures, we figured out that an avian-like open or at least semi-open nest evolved here in this clade that contains Oviraptors, Triodontids, Deinonychus, toothed stem birds, and modern birds. This clade is often called Eomaniraptora. Some call it Panaraptora. And the basal-most representatives are Oviraptors. So we started to analyze Oviraptor eggshells, 3 different kinds, all assigned to Heyuania huangai from 3 different localities in China. We analyzed these eggshell fragments and started with what is called an organic phase extraction that eventually leaves us with an extract that in theory contains any sort of traces of fossilization products of potentially present eggshell pigments. We've removed all sorts of minerals from our extracts to make sure that salts or minerals are not going to mess with our analyses for pigments. And eventually we've subjected our fossil eggshell extracts to a method called reversed phase high performance liquid chromatography, electrospray ionization, quadrupole time of flight mass spectrometry. Sounds really complicated, but it's super simple. Mass spectrometry eventually works like a super sensitive molecular scale. So once you analyze your standards for biliverdin and protoporphyrin and get the exact molecular weight, you can basically analyze your eggshell extracts to figure out if corresponding exact masses identifying biliverdin or protoporphyrin are present. And here came a huge surprise, because we found both biliverdin and protoporphyrin present in all 3 eggshells. Generally, we found only trace amounts of pigments preserved, and the quantities of biliverdin and protoporphyrin differed, relatively speaking, in all 3 eggshells. Still, that was good enough to allow us to come up with the first color reconstruction for Oviraptorate eggshells, as shown here. We also analyzed associated sediment samples and didn't find any evidence for biliverdin or protoporphyrin, suggesting that we're dealing with some sort of original residues of egg color pigments and not with any external contamination. We've added this one data point here in our Archosaur tree of life. We have crocodiles here on the top and birds here at the bottom. And as you can see, when we try to understand how egg color evolved, this one data point eventually raises more questions than it provides answers. If we look at how egg color could have evolved just by adding this data point with all these blank gaps in between, there are 2 different possibilities. Egg color could have had a single evolutionary origin in the clade Eomaniraptora, or egg color could have evolved multiple times independently, once in Oviraptors and at least one more time in modern birds, potentially multiple times. We know that the egg fossil record is somewhat patchy, and there's definitely a bias towards these small, carnivorous, raptor-like dinosaurs. However, all taxa that I've listed here in this phylogeny have fossil eggshells preserved that can be assigned with quite some confidence. For some of them, we know the nesting modes, and for all of them, we can potentially figure them out based on the egg-shell porosity. However, there is a problem when we try to close these gaps to really figure out how egg color evolved. And that is primarily that some of the eggshells that are included here are incredibly valuable and unique. While we have lots of different eggshells associated with embryonic material that can be assigned to oviraptors, we do not have that much eggshell material for other kinds of dinosaurs. For example, the Deinonychus eggshell that I'm including here, this one, is unique. There's only one Deinonychus eggshell in the world. So obviously, we could not analyze it for eggshell pigments by powdering down our eggshell sample and doing an organic phase extraction. So we had to come up with a new method that is non-destructive, meaning that it doesn't require us to destroy the fossil. The method we're using now is called Raman microspectroscopy. Raman spectroscopy is a wonderful non-destructive tool. Eventually, we shoot a green laser on our eggshell sample. Molecules in our eggshell sample get excited by the green laser light, and their bonds start to vibrate in very characteristic ways corresponding to their chemical environment. Eventually, we can pick up these signals in form of peaks and valleys in our Raman spectra. Every peak represents a characteristic molecular unit, a characteristic molecular vibration, and the information is encoded here on their position on the x-axis. We can fingerprint the specific Raman signature of biliverdin and protoporphyrin and eventually compare it to our fossil eggshells to see if we find evidence for our pigments. But there's another problem. There are more things in eggshells that preserve than just pigments. So when we're not performing an organic phase extraction that eventually just gives us the pigments, we have to deal with all kinds of organic compounds that are present in the eggshell. If you remember my slide from earlier, we have all sorts of proteins present in eggshells as well. They act as some sort of organic skeleton. Proteins fossilize as well, and they are transformed into so-called protein fossilization products. Here we're seeing some protein fossilization products extracted from fossil eggshell. And as you can see here, we're definitely dealing with a lot of chemical transformation and only small amounts and residues of what they originally constituted in terms of structure are preserved. The problem with protein fossilization products is that the way they fossilize, they resemble to a certain extent our egg color pigments. And I'm showing you this right here. When we look at the fresh modern protein represented here by this tripeptide, we're dealing during fossilization with the transformation into so-called nitrogen, sulfur, and oxygen heterocyclic polymers. We form these nitrogen heterocyclic units that, as you can see here, resemble quite closely the pyrrole units that make up our tetrapyrrole pigments. To be sure that the Raman signatures we interpret are truly representing our egg color pigments and not molecularly similar compounds, we had to play a trick. We fingerprinted eggshell protoporphyrin, here shown in red, eggshell biliverdins, the spectrum is here shown in blue, and fossil dinosaur bone, here this is an Allosaurus bone, that contains only protein fossilization products. What we see here is that many of the peaks sort of overlap, but there are some peaks that are quite characteristic for the different kinds of compounds. Here for biliverdin and here for protoporphyrin. Based on these characteristic peaks that are not present in protein fossilization products, we came up with a characteristic set of bands that allow us to positively identify biliverdin and protoporphyrin even when there are protein fossilization products preserved in the very same eggshell. We basically have these band sets and we require a positive identification of all the characteristic bands in our fossils. And this is exactly what we did with all the dinosaur eggshells that I've just shown you before. We can get spectra that eventually tell us something about the total composition of organic compounds in our fossil and about the presence of biliverdin and protoporphyrin. But we can also map out characteristic bands for certain compounds, like, for example, protoporphyrin. As you remember, protoporphyrin is the only pigment that can cause spots and speckles. And so we're particularly interested in the distribution of protoporphyrin. So we've mapped out protoporphyrin over the surface of our different eggshells. Here we have our oviraptor eggshell, a couple of cheirodontid eggshells, Deinonychus eggshell, toothed stem bird, and then we have emu and chicken eggshell, sort of as a modern comparison. Below you see the color reconstruction. We found evidence for spots and speckles in all our fossil samples. And eventually we got different modes of background coloration present. And all of them are shown here, sort of like with a plausible range reconstructed for our dinosaur eggshells. That is incredibly exciting. But as you remember, there is the possibility that dinosaur and bird egg color may have evolved still independently. So how are we going to test for that? We're being smart about it. Here we're looking at cross-sections of our fossil and modern eggshells. Again, the ones that correspond to the eggs shown above. As I mentioned, bird eggs have a very characteristic mode of incorporating pigments across their vertical cross-sections. So you can see here, biliverdin extends into the deeper layers of the eggshell with highest concentrations in the cuticle, while protoporphyrin is always constrained with the highest concentrations just to the waxy cuticle. We wanted to figure out if fossils, specifically dinosaurs and stem birds, incorporated their pigments in the exact same way into the eggshells, which would be quite convincing evidence for non-avian and avian dinosaur egg color being homologous, so basically being the same thing. Here we see in red protoporphyrin and in blue biliverdin distributed across the thickness of the eggshell. For modern birds, here emu and chicken, and here for our fossil representatives. And as you can see, the mode of incorporation is pretty much the same. We have a little bit of alteration based on fossilization processes, but in the end, we can conclude that dinosaur and bird egg color are homologous. Still, you may want to ask, why are the dinosaur eggs that we've seen before not colored anymore? And that is a question that we've also asked ourselves. Of course, there are only trace amounts of unaltered pigments preserved, but why are there no clear color impressions left? Is there anything that is overprinting this color? We have analyzed protoporphyrin from eggshells and biliverdin from eggshells and subjected them to experimental fossilization conditions. And what you can see here is basically the color change of the pigments during these oxidative fossilization conditions. Eventually, we end up with organic residues that resemble pretty much what we see in our fossil eggshells in terms of coloration. We have trace amounts of our unaltered pigments, protoporphyrin and biliverdin, still present here, but the majority of pigment has been transformed, and most of them in quite different ways, giving us a range of different fossilization products. As you remember, small differences in the molecular structure can yield fundamental differences in the way that these pigments generate color. And because we have here like a mix of different molecular versions of different fossilization products of our pigments, we have also a mix of different colors that eventually yield overall a brown color impression. This mode of discoloration is also often observed in museum eggshells that are exposed for like a long amount of time to air or to light. And the kinds of alterations that we see in museum eggshells are very much comparable to what we see in our dinosaur eggshells. Now we can fill in the gaps in our evolutionary scenario. And as you can see here, based on the data we have now, we can infer that egg color had a single evolutionary origin in Eomaniraptor and dinosaurs, coinciding with the evolution of open nesting habits. I think it is quite remarkable that the diversity in terms of color and pattern that we see in modern bird eggs is mirrored by dinosaur eggs. We have all kinds of color shades reconstructed here based on our pigment signatures in the spectra, and we find evidence for spots and speckles in almost all fossils that we've analyzed. When we published these results in 2018, we got a very important and interesting question from an independent team working in Belgium. They basically asked, the method that you're using, does it really detect egg color, or do you just detect actual pigmentation? What does this really mean? So we can have a white eggshell that is macroscopically, visually perceivable, not colored, and still contains teeny tiny trace amounts of biliverdin and protoporphyrin. We need a certain minimum concentration, a certain minimum amount of pigment to generate a visually perceivable color impression. So we went back to the lab and we analyzed a set of bird eggshells, some of them barely colored, some of them white, some of them brightly colored, to figure out what the detection limit of our method is in terms of quantity of pigments. When do we get a positive pigment identification? We found that we get a positive pigment identification for pigment quantities that exceed the amount needed to generate a visually perceivable color impression. So eventually, our method really detects egg color and not just eggshell pigmentation. With this information, we could be quite sure about our color reconstructions in fossils. Eventually, we want to use the color information that we get for fossils to learn something about the behaviors of extinct animals. I think this is one of the most exciting parts here, because eventually we're using very minimal and very reduced information preserved in form of fossils to learn something about the lifestyle, about the nesting, about the parenting of long extinct animals. In modern ornithology, there are very good correlations between certain nesting and parenting strategies and characteristic egg colors. And so here we're going to interpret what we learned from our egg colors. Blue egg colors, like for example, present in oviraptors, correspond primarily to paternal care, meaning that the fathers would guard the nests. Eventually, we find blue egg colors in modern birds, primarily in areas that have a relatively high temperature and humidity during the reproductive season, giving us a nesting environment reconstructed like here in this figure for our oviraptor. Brown egg colors, like for example present in some of our triodontids, indicate generally maternal nest care, meaning that the mother would guard and brood the nest. And we find them in areas with relatively low temperatures and humidity during the reproductive season, giving us a nesting environment reconstruction like shown here. Egg spots and speckles are particularly exciting because they tell us so much about the nesting ecology and interactions of different species during the nesting, brooding, parenting part. So generally, individual egg recognition is required, especially in so-called communal nesting, in communal nests. Communal nests are basically nests where multiple females lay their eggs in one shared nest. There is the need to identify their own eggs based on characteristic patterns of spots and speckles against all the other eggs to be sure that they identify their own chicks after hatching. Also, spots and speckles are particularly important when it comes to nest and brood parasites. If you think about cuckoos that lay their eggs into the nests of host birds, they start some sort of evolutionary arms race, quite often forcing the host bird to evolve very sophisticated patterns in terms of spots and speckles to distinguish their own eggs from nesting parasites. All this eventually tells us that dinosaur reproductive biology was much more complex and much more bird-like than we think for the moment. And so there is a lot of future work to be done. At this point, I'd like to thank you very much for your attention. And I'm happy to answer any questions.
All right, great. Thank you so much. That was fantastic. And I know we had a lot of people listening in. So thanks to everybody who joined us today. We have a couple of questions that have come in already, Jasmina, and I will pose those to you. Unless you
Awesome.
can, I don't know if you can see those on the chat, maybe not, but I think probably you can't. So let me pose those. But anyone else who has a question, please use the chat function right now to just send your question and we will pass it along. So the first question is a bit of a technical one, and it actually comes from our collections manager of mineralogy and meteoritics here at the Peabody. And the question is, which calcium carbonate polymorph makes up the mineralized part of the eggshell?
So for most eggshells, it's calcite. And then, so like for all bird eggshells, it's primarily calcite. So it's basically, well, yeah, it's a particular kind of like variety of calcium carbonate. Then we find in bird eggshells, right below the cuticle, there is like a layer that is called the mineralized cuticle, and that contains calcite and apatite. So it's like a calcium phosphate, so a completely different kind of mineral, but it's only present in trace amounts there. And then if we kind of like go like outside of birds and dinosaurs, in turtles we have aragonite as key mineral that makes up the eggshell. So there's quite a bit of quite a bit of variability. Yeah.
Perfect. I think he had a follow-up question, but I think you actually answered it just there. So yeah,
Okay.
that's fantastic. Okay, we've got a couple other questions here. Hi, Jasmina. This is Mark Hauber. So impressed with your research. Do you already have evidence of brood parasitic dinosaurs based on spotting / color dissimilarity?
Yeah, hi Mark. Great that you listened. He's like one of the world's leading experts on egg color in birds. So yeah, that's fantastic. Hey. So I don't think that we have like for now any key evidence for brood parasitism in dinosaurs. There are a couple of clutches, where we have like eggs of a particular type of dinosaur and then like eggs of like other animals. Like for example, quite often we find like crocodile eggs associated with some dinosaur eggs. But we think that this is more like, something that is like related to like the diagenetic compacting of different layers. So yeah, we totally have to go there at some point, look at spots and speckles and figure out if we really had like brood parasitism in dinosaurs. I wouldn't be surprised at all, honestly.
All right, great. Thank you. Here's one from Claudia Gros or Gress. How do you determine paternal versus maternal nest brood behavior?
Okay, yeah, so I'm not doing that myself. There is a wonderful paper, and I can share the link later on if you guys are interested. We can probably do this on Twitter also. There's like a wonderful ornithological paper where the authors looked at what kinds of behaviors are associated with certain egg colors, and they came to the conclusion that brown egg colors are more commonly associated with maternal nesting behaviors, and that blue egg colors are more commonly associated with paternal nesting behaviors. And so I'm really just using their wonderful research across like, I think they use like lots and lots of species of birds, like 250 species of birds, something like that. Yeah.
Cool. Wow, fantastic. All right, here's one that says, excuse me, I'm only 7. I think you answered this question already, but why are my eggs from the supermarket white?
So that's actually a really good question. Yeah, so like, when you like shop like chicken eggs from the supermarket, you can get like the brown ones, or you can get the white ones. And if you go to like a really fancy farmer's market, you can even get green ones. They are like, it's kind of a chicken breed that's related to the jungle fowl or something like that, a puche fowl, and they lay like green eggs. So many of the chicken eggs in the supermarket are white, and it's simply based on customer preferences, because people want their chicken eggs to look as clean as possible, right? Because it's like food, and people like, want to be sure that it's like super like clean, that there are no feathers sticking to it. And this is why many chicken eggs are white, because chickens were bred in that particular way. But all these kinds of eggs are equally tasty. So.
Excellent. Thank you. All right, so this is from Erin. Do we know enough about bird and dinosaur visual perception to say what parts of the electromagnetic spectrum they see if they are similar?
That is a really, really interesting question. So as you may know or not, some birds are able to kind of like see UV, certain like UV wavelengths. And we're mammals, so we're like really sad in our visual setup compared to birds. So for dinosaurs, just based on the phylogenetic inference, it could very well be that many dinosaurs, well, most likely dinosaurs were able to perceive certain UV wavelengths as well, see UV as well. But yeah, there's no direct evidence, of course. So like that all remains subject to phylogenetic inferences and speculation.
Excellent. All right, we have one for, not sure who it's from, but it says, does the large number of dinosaur eggs say anything about survivability or maternal bonding?
Ooh, okay. So generally, like there are some dinosaurs, like for example, like sauropods, long-necked dinosaurs, that like dump like dozens of eggs in a nest. And they would be considered so-called like R strategists, right? So they produce like lots of offspring. And then generally what we see in like modern animals that are so-called R strategists, they would basically have like a rather low survivability. They would have like a very high mortality of like young chicks that hatched. And then there are other dinosaurs, especially on the branch leading towards birds, where we are like in carnivorous dinosaurs and theropods, where we have definitely like a shift towards K strategists. So basically animals that produce fewer offspring, but there is more parental investment. So like the hatchling mortality would be much lower. So to a certain extent, I guess the number of eggs is representative of the, let's say like hatchling mortality, but this is all based on inferences based on extant taxa.
Cool. All right. Mark, by the way, says thanks so much. And he had asked a question earlier. Here's one from Karen. Did dinosaurs always nest on the ground, or any evidence of nests in trees?
Yeah, that is super interesting, right? So for the moment, we only have kind of like clutch. Well, so if you look at the fossil record, right, everything is sort of like embedded in rock. And so for now, like with the nesting structures that we sort of like reliably reconstructed, we only found nesting structures associated with ground nesting. And we know based on the porosities of these eggshells that most of them like even like oviraptor eggs and some troodontid eggs. So like, you know, dinosaurs that build sort of like open nests, they buried half of the egg underground. But it's really hilarious to think about. If we have here like an emu egg, that would be basically half underground and half sticking out. And so, yeah, like, I mean, I could totally imagine that there were dinosaurs building nests on some sort of like tree-like structures or on rocks and cliffs. But then we have to think again about the fossilization potential of these things. And I mean, I guess there is quite a bit of a taphonomic bias that prevents these kinds of nests from being recorded in the fossil record. Because to become a fossil, you need a really high sedimentation rate. And so you don't necessarily have that when your nest is not localized on the ground. So yeah, we don't know.
Fantastic. We're down to the bottom of our chat list of questions. I think this might be a logical time to bring the presentation to an end. Jasmina, it's really an honor to have you with us today, sharing your research and your work. We can't wait to see what comes next. I know there's publications that are going to be coming out, really soon, and I encourage those of you who are familiar with accessing the scientific literature to follow Jasmina's work there. But I just want to, on behalf of the Peabody Museum, I want to thank you so much for doing this with us today. This is a virtual gallery talk, and I think we'll figure out how to do more of these. So I'll just say to everybody out there, let's give Jasmina a round of applause, even though